Okay, so a word about myself. I'm at the UNM Health Science Center. My group is the cytometry group, and I'm associated with the pathology department in the cancer center. I'm also a co-founder of the company called IntelliSite, which has been acquired by Sartorius. Um, in this particular project, the inhibition of cellular uptake to treat bacterial infections, there are four main contributors. Myself at UNM, working on the discovery side, Mark Haynes at UNM, and two collaborators at Ball State University, Susan McDowell, who's our expert in microbiology, and Robert Samuelson, who is our expert in synthetic chemistry. So the way that we look at this project is in terms of the innovation. We have already identified molecules that block bacterial uptake into target cells. And so we think about these as non-antibiotic therapeutics. They could limit host cell invasion, and therefore they could improve the extracellular antibiotic efficacy. They could increase clearance by immune cells. They could break the cycle of relapsing infection, and they could reduce infection-related morbidities and mortality. In terms of the background, one of the areas that we've worked in previously is a multiplex discovery technology, which means that we can look at multiple targets simultaneously, that we've applied to low molecular weight GTPases. And with Dr. Angela Wander-Ness, there are 5 US patents. There's also a startup company involved in clinical applications in cancer. That's called Revere. So these low molecular weight GTPases control receptor signaling, membrane trafficking, cytoskeleton, and nuclear uptake. And during our discovery process with these multiplex screening tools, we identified a molecule that is selective for a specific GTPase called CDC42, which controls cytoskeleton. And so Ball State was interested in the relationship of bacterial invasion, infectivity, and cytoskeleton because bacteria have to go through, transit through the cytoskeleton on their way to becoming infective. So these complementary research activities were brought together here, and the research is now protected by 4 US patents that are administered by the UNM Innovation Center. The application for this technology is that we understand that antibacterial agents typically induce resistance, and systemic delivery represents a high barrier to success for new antibacterial agents. So what we're focusing on specifically then is a host-directed approach. So we're directing these molecules at the host, the target cells in the host, rather than the bacteria. And the idea is that the blocking uptake, we can limit infection to improve clinical outcomes by breaking the cycle of recurrent infection. The specific types of models that we're interested in are pulmonary infections and pulmonary administration, which are intended to be systems where we actually reduce the systemic burden of the new molecules that we're administering. The need for this technology is that it's common. People understand that antibiotic resistant strains are emerging in the clinical arena. And there's also a limited efficacy of these first-line antibiotics, which are available against intracellular populations of the bacteria, because now you have to get molecules into cells and into the bacteria inside of cells. So there is a market potential, which is related to the morbidity and mortality. And there are a number of situations where there are infections, some of them in the lung. So the types of infections that we're talking about are prosthetic joint infections, where with 600,000 procedures annually, there's an estimated risk of infection about 2%. Likewise, infective endocarditis, where there's an annual incidence in the US of 10 to 15,000. But most importantly, as a starting point, is cystic fibrosis, which is a disease of the lung. There are about 30,000 people living with cystic fibrosis and about 1,000 new cases annually. So we've done proof of concept experiments with these molecules that block the uptake of bacteria. And so there are two panels. These experiments can be done in ways which have sufficient throughput to analyze new molecules. And what we can measure by flow cytometry is the percent of cells that have internalized bacteria in the presence of this compound on the left, and with an IC50 of about, an EC50 of about 5 micromolar. And we can actually count the bacteria that are taken up in individual cells, comparing controls, which are labeled PEG, the compound itself, and the compound which is washed out. So this is a compound that we have protection of composition of matter. And we've also developed improvements to this specific compound in this field of use. So the status of the project is that there are 4 patents issued for composition of matter, field of use, and improvements which are already in hand. The intellectual property is managed by the UNM Innovation Center. We know how these compounds work. They specifically work by inhibiting in a selective way the cytoskeletal protein CDC42. We already know that co-treatment of in an infection model with the antibiotic rifampicin reduces intracellular Staph aureus infection. And we have seen commercial interest in this project. What's worth knowing at this time is that I am now emeritus, and so I personally have time to devote to this project in the next phase of my career. The plans for this project are to secure seed funding, to expand proof of principle, animal model system studies. So these would be pulmonary infections with pulmonary administration. We've begun preliminary discussions for potential affiliation with the New Mexico Startup Factory for SBIR and SGTR. We also have been in discussions with potential partners for pulmonary administration of the compounds in animal models, and we have the capacity to engage in further improvements in the activity and physiological properties of the molecules once we have defined their proof of principle in animal models. Thank you for attention.
Thank you, Larry. Over to our shark panel.
Hey, Larry, this is Mike. Couple things I would typically like to see in a presentation like yours. One would be a high-level product development plan overlaid with the best understanding of what the costs will be to bring the technology to market, assuming that you do it yourself or through a partner. And then the second thing would be regulatory pathway as best you understand it today. Can you just comment on both of those kind of topics just to give us a sense of your thoughts at this stage?
So say those two things again.
So the first will be your anticipated time frame and major development milestones to get the product to market, and then cost around that. Let's start with that one.
Yeah. So in terms of the milestone, from my perspective, the first milestone is demonstration of efficacy in an animal model. And I'm estimating that pilot studies, we should have some data, sort of a go/no decision with somewhere between $50,000 and $100,000. We haven't priced that out specifically because I think there are going to be different regulatory issues for pulmonary administration versus systemic administration, and frankly, those need to be evaluated in more detail. So we're talking about the time after we get seed funding, and that should be something. So let's say seed funding is 6 months. Pilot data, go, no go, is probably another 6 months. And, after that, it's going to depend upon efficacy and what we learn about safety of these molecules administered at the types of concentrations delivered in the lung, where the systemic concentrations are expected to be, orders of magnitude lower than what we actually apply in the lung. So, that's sort of a high level perspective, and we actually need some data in order to fill in some more details, because there will be bifurcation points depending on what we see.
Okay.
Larry, this is Joey. I just want to say, I think the cystic fibrosis aspect of this drug is quite interesting to me. We're actually looking at a company that I think could potentially be a partner of yours. I was going to ask you a question about the potential side effects of the drug, but I guess that's pretty early.
Say it again.
I guess my other question would be, do you have a sense of what the dosage amount would be if it was delivered through inhalation?
Yeah, we're talking about milligrams.
Milligrams? Okay.
Yeah.
Hey, Larry, this is Mike. Just another question on what corporate interest have you seen thus far? I think companies maybe we've seen and worked with oftentimes will follow a kind of a corporate venturing pathway. What does that landscape look like for you at this point?
Yeah, so the only connections that we've made have been early startup connections through the Innovation Center, and the first approaches were companies that were, not not very far along themselves and not being a good fit. So this is actually the first public presentation of this concept, and I appreciate the input that you're providing about what I need to learn in order to be effective in moving this forward. But we haven't gotten beyond today. I mean, what you're hearing today is the first time this has been out in public, which is not meant to be an excuse, but I'm here to learn from the experience.
Got it. No, thanks for that clarification. And then last question I had for you, there sounds like there's multiple patents, 4 issued, maybe 1 provisional. It's a little unclear how this IP relates specifically to your plans for this technology. I mean, it sounds like there's potentially another startup, and have they licensed some of these patents?
Yeah. So altogether, let me do a recount. There are 5 patents that are with Angela Wunderger-Ness that we did discovery together. And there is a different molecule which is going into cancer directed at a different target. So we did a multiplex screen. We've screened, I want to say dozens, literally dozens of different GTPase targets in the screen. And so there's a platform which has a patent. Then there are applications in cancer. So that was the first 5. One of those patents in the first 5 was for this molecule itself. And then there are 3 additional patents with Ball State. So the composition of matter is held at UNM. The field of use and the improvements are held jointly with Ball State. So there are 3 in those areas. So this field at UNM, I mean, I'm talking about 8. There are altogether more than 10 patents in this area of GTPases. The one common patent here is the composition of matter for the molecule that's called ML41, but there then are field of use in infections as opposed to cancer, which are separate. Does that help clarify? Or is it too complicated?
Well, somewhat. I mean, this is definitely, there's a lot there. So I think that answers the question at a high level for sure.
Yeah, yeah.